Mr. Taylor has been feeling tired and weak lately, so he's going to see Dr. Endicott. Dr. Endicott notes that Mr. Taylor has a family history of many different types of cancers. His father died of a brain tumor. His brother died of pancreatic cancer. His sister is fighting breast cancer. And his son was diagnosed with bone cancer last year. Dr. Endicott performs some lab tests and learns that Mr. Taylor has colon cancer. The colon is the first 6 feet of the large intestine. Because Dr. Endicott caught the cancer early, Mr. Taylor has a good chance of surviving. Mr. Taylor wants to know why so many people in his family have developed some type of cancer. Looking at the family history, Dr. Endicott diagnoses the family with Li-Fraumeni syndrome, a hereditary disorder characterized by familial multiple tumors. The tumors are caused by a mutation in the p53 gene. A mutation is a change in an organism's DNA. The p53 gene codes for a protein that regulates cell division. Though the exact mechanism isn't known, the p53 protein senses problems in the cell's DNA. When the protein detects DNA damage, specifically incorrectly paired or mismatched bases, it tells the cell not to divide until the DNA has been repaired. If the DNA can't be fixed, p53 tells the cell to commit suicide, so the damaged DNA is not passed on to daughter cells. When p53 is mutated, the protein can't do its job, and mismatched base pairs may not be caught and fixed. Any mutation or change in the DNA is passed on to all daughter cells. With a mutant p53 protein, mistakes in the DNA are not repaired before cell division. More and more mutations accumulate with each cell division. Eventually, the genes that keep cancer from occurring will be mutated. Depending on which genes are mutated, different cancers occur. Li-Fraumeni syndrome shows why it's important for cells to detect changes in the DNA during replication. Mr. Taylor's father was born with a mutation in the p53 gene. Therefore, his cells were unable to detect changes in his DNA, and a brain tumor formed. The p53 mutation was present in his sperm cells, so he passed the mutation to his children. Mr. Taylor then passed it to his son. One way mutations can occur is during DNA replication. During DNA replication, all of the organism's DNA is copied. For humans, around 3 billion base pairs have to be copied. Because changes in an organism's DNA are rarely beneficial, it's critical for DNA replication to be very accurate. The structure of DNA provides a perfect mechanism for ensuring the DNA replication is very accurate. Also, normal cells have proofreading mechanisms to catch errors in the DNA. In this animated presentation, we'll look at the details of DNA replication. Then we'll investigate how cells ensure the accuracy of DNA replication. As we've seen, changes in an organism's DNA can be very harmful. Because of this, accurate DNA replication is very important. The structure of DNA provides a very accurate mechanism for DNA replication. DNA is a double helix, with the 2 strands in an anti-parallel orientation. The backbone of each DNA strand is made up of phosphate groups and deoxyribose sugars. Each nucleotide unit also contains a nitrogenous base. Because adenine always pairs with thymine and guanine always pairs with cytosine, a single strand of DNA can serve as a template for the other DNA strand. Watson and Crick first proposed this replication strategy when they determined DNA structure. However, biologists still considered three possible models for how replication occurs. In the conservative model, each time the DNA is replicated, the parent double-stranded DNA molecule, shown in dark blue, stays intact, and an entirely new double-stranded molecule, in light blue, is made. After 2 rounds of replication, there are 3 entirely new DNA molecules, along with the original double-stranded DNA molecule. In the semi-conservative model, the strands in the parent molecule separate, and each strand makes a new double-stranded molecule. The same process occurs in the next round of replication, yielding 2 molecules with one of the original parent strands and 1 new strand, plus 2 molecules containing none of the original DNA. This is the model Watson and Crick originally proposed. In the dispersive model, each new daughter strand is a mixture of original and new DNA. In the second round of replication, each DNA molecule contains some of the original DNA. In 1953, American biologists Matthew Meselson and Franklin Stahl devised an experiment to determine which replication model was correct. They grew cells of the bacterium E. coli in the presence of a heavy nitrogen isotope. The heavy nitrogen was incorporated into the bacterial DNA during replication. Next, they switched the bacteria to media containing a lighter nitrogen isotope. Since bacteria divide about once every 20 minutes, the first round of replication was complete after 20 minutes, and any newly synthesized DNA was made with the lighter nitrogen. After 40 minutes, the second round of replication was complete. To determine which nitrogen isotopes each double-stranded DNA molecule contained, they extracted the DNA from the bacteria and separated the DNA molecules by density. DNA made with only the lighter nitrogen isotope stayed near the top of the liquid, while DNA with only the heavier nitrogen isotope went to the bottom. DNA molecules containing some of each type of nitrogen ended up in the middle. The DNA extracted after 20 minutes was of intermediate density. Which model do the results rule out? Click Submit to see if you're correct. Sorry, that's not right. The conservative model predicts that the first round of replication will yield the original DNA composed of only heavy nitrogen and a new DNA molecule composed of only light nitrogen. If this model were correct, Meselson and Stahl would have seen a band for the heavy DNA and a band for the light DNA. Since they saw one DNA band of intermediate density, the conservative model cannot be correct. The DNA extracted after 40 minutes had a light band of DNA and a band of intermediate density. Select the replication model supported by the data. Click Submit to see if you're correct. Sorry, that's not correct. The dispersive model predicts that each DNA strand will be a mixture of heavy and light, and that all of the DNA molecules will have roughly the same density. The semi-conservative model predicts that the second round of replication will yield 2 DNA molecules composed of only light nitrogen and 2 molecules composed of 1 heavy and 1 light strand. Since Meselson and Stahl got 1 band corresponding to the light DNA only and 1 band for the combination of heavy and light, the semi-conservative model is correct. While the basic concept behind semi-conservative DNA replication is elegantly simple, the actual biochemical mechanism is quite complicated. Replication begins at special sequences of DNA called origins of replication. Bacterial chromosomes have one origin of replication. Eukaryotic chromosomes, which are much larger and have more DNA to replicate, have many origins. Proteins that initiate replication bind to the origin and separate the two DNA strands, creating a replication bubble where the new DNA strands are being synthesized. Replication occurs in both directions. Since eukaryotes have multiple origins of replication, they have multiple replication bubbles moving simultaneously. The bubbles eventually collide. Each end of a replication bubble has a replication fork, a Y-shaped region where new strands of DNA are elongating. At the base of the replication fork, the helicase enzyme unwinds the helix and separates the DNA strands. Enzymes called DNA polymerases add the nucleotides to the growing DNA strand. Nucleotides align with their complementary bases one at a time on the template DNA once DNA polymerase is bound. As each monomer unit is added to the strand, two phosphate groups are released. The energy released in this reaction drives DNA synthesis. DNA polymerase can only add nucleotides to an existing DNA strand. To initiate replication, a short stretch of RNA called a primer is needed. RNA is similar to DNA. The major difference is that RNA is made of ribose sugars, while DNA is made of deoxyribose sugars. DNA polymerase adds nucleotides to the RNA primer. Once replication has begun, the RNA primer is replaced by DNA versions of the nucleotides. The new strand of DNA is elongated 5' to 3'. The fact that DNA strands run in opposite directions presents a problem at the replication fork. As the DNA is unwound, one strand can be elongated in a single piece in the same direction as the replication fork is moving. This is called the leading strand. The other strand, the lagging strand, has to be elongated in pieces in the opposite direction as the movement of the replication fork. The pieces are known as Okazaki fragments. The overall direction of DNA elongation is in the direction of the movement of the replication fork. Now that you've seen the components, try building your own replication fork. Drag the components into the correct locations on the replication fork. You only need to use 2 of the DNA strands, but you may use some of the other components more than once. Click Submit to see if you're correct. Sorry, that's not quite right. Helicase unwinds and separates the DNA at the base of the fork. A primer is needed to begin replication. DNA polymerase adds nucleotides to the growing strand in the 5' to 3' direction. This means the leading strand is elongated in one piece, while the lagging strand is composed of small pieces of DNA, the Okazaki fragments. As we've seen, changes in an organism's DNA are not usually beneficial. The cancers in Mr. Taylor's family are the result of a mutation in the p53 gene, which normally senses mismatched base pairs in a cell's DNA and halts cell division until the DNA is repaired. Since the members of the Taylor family have a mutant version of the p53 gene, their cells continue to divide even when the DNA has errors. The DNA errors are passed on to the daughter cells. Because of the mutant p53 gene, more DNA changes occur with each cell division. Depending on which genes are mutated, different problems arise. Mr. Taylor developed colon cancer, while his brother developed pancreatic cancer. As we see with the p53 mutation in the Taylor family, once DNA is altered, the changes are passed on from generation to generation. Because faithful DNA replication is so important, organisms have a number of ways to ensure replication accuracy. The first defense against base pair changes caused by replication errors is DNA polymerase. This enzyme adds the bases to the growing DNA strand. DNA polymerase rarely makes mistakes, but for extra protection against errors, it also acts as a proofreader. If the wrong base is added to the growing DNA strand, DNA polymerase senses the mismatched base. The polymerase backs up and fixes the mistake. The proofreading activity takes advantage of the specific base pairing nature of DNA. Since A pairs with T and G pairs with C, mismatched bases are easy for DNA polymerase to recognize. Considering how important faithful DNA replication is to an organism, it isn't surprising that many other proteins are involved in ensuring accurate DNA replication. For example, the p53 protein senses mismatched bases and tells the cell to stop dividing until the damage is repaired. If the damage can't be repaired, the p53 protein signals the cell to commit suicide. To the organism, killing one cell with an error is less harmful than allowing the cell to live and generate daughter cells with the same error. Li-Fraumeni syndrome is a good example of how harmful it is to have a mutation passed from generation to generation. If the p53 mutation had been caught before being passed to Mr. Taylor's father, this family would not have developed so many cancers. Besides DNA replication errors, there are many other causes of DNA mutations. Ultraviolet light, hazardous chemicals, radioactive emissions, and X-rays all have harmful effects on DNA. DNA repair enzymes protect our skin from damage due to overexposure to the sun. More than 50 different types of DNA repair enzymes have been identified that monitor and repair damaged DNA. Most of the repair enzymes take advantage of the base pairing of DNA by using the other strand as a template. For example, in excision repair, a stretch of damaged DNA is cut out and the other strand is used as a template to fill in the gap. The sun's ultraviolet rays cause DNA damage in human skin cells. The UV light causes adjacent thymine bases to become covalently linked. This is called the thymine dimer. The covalent bond causes the DNA to buckle, which leads to errors in replication. Excision repair proteins cut out the section of DNA containing the thymine dimer. DNA polymerase uses the other strand as a template to fill the gap. Because of the structure of DNA, the basic mechanism for DNA replication is elegantly simple. One double-stranded DNA molecule can be used to produce two identical molecules of double-stranded DNA by copying each individual strand independently. While the basic semi-conservative model of DNA replication is simple, the biochemical mechanism is much more complicated. Replication begins at specific sequences of DNA, the origins of replication. Prokaryotes have one origin of replication, while eukaryotes have many. At the base of the replication fork, helicase unwinds and separates the DNA strands. A short stretch of RNA called a primer is necessary to initiate replication. DNA polymerases then add nucleotides to the growing strand. The new strand of DNA is elongated in the 5' to 3' direction. Since the two strands in a double helix are antiparallel, only the leading strand can be made continuously. The lagging strand is elongated in small pieces called Okazaki fragments. Changes in an organism's DNA are usually not beneficial, so it is important for DNA replication to be very accurate. Any change in the DNA is passed on to all daughter cells. The base-pairing nature of DNA provides a mechanism for faithful replication, but there are also other safeguards. DNA polymerase acts as a proofreader, looking for mismatched base pairs as the DNA is replicated. Scientists have identified over 50 enzymes involved in ensuring accurate DNA replication, including the p53 protein. There are many other causes of DNA mutations besides DNA replication errors, and cells have mechanisms for dealing with them. Excision repair cuts out and repairs stretches of DNA with errors in only one strand, including thymine dimers caused by the sun's UV rays.